everyone. My name is Willa. I'll be a rising senior at Lowell High School, and this summer I was very lucky to work with Michael Burgess and Eugene Huang at the Shannon Lab. And the title of my project is Restoring NF1 Gap Activity Impairs Blast Colony Formation in NF1 Deficient Murine Acute Myeloid Leukemia. So first, acute myeloid leukemia, or AML, is a form of blood cancer where there is an overproliferation of these immature white blood cells that crowd out these healthy blood cells. And that's problematic because it can lead to anemia and difficulty fighting infection, and also the cancer can spread to other organs. So to investigate this overproliferation, we look at the RAS protein, which signals through many signaling pathways, and one thing that it regulates is cell proliferation. So RAS is on when it's bound to GTP, off when it's bound to GDP, and there are these proteins called gaps that turn off RAS, and the gap that I studied was neurofibrillin 1, or NF1. So we've previously shown that if we delete NF1 in mice, we can induce acute myeloid leukemia. And in these NF1 deficient AMLs, if we give them a drug that inhibits the activity of a signaling molecule downstream of RAS, it helps for a little while until we get drug-resistant clones that develop. So what we want to know is NF1 loss required for AML maintenance in drug-sensitive versus drug-resistant clones? And knowing this will tell us if RAS signaling is still being used, or if there's a different signaling pathway that is allowing for this drug resistance. So this project, we are going to restore NF1 gap activity to AMLs and observe their effects on growth. So how we did this was we designed DNA constructs that expressed the gap part of NF1 called NF1-GRD. We tagged it to a green fluorescent protein. We also designed a control construct that was also tagged with GFP. We put these constructs into virus that we infected into primary leukemia cells. We sorted the ones that were green so we knew that they were expressing our constructs. Then we plated them in methylcellulose colony assays, either in the presence or absence of growth factors. We grew them for a week, then we counted colonies. So our results show that in a drug-sensitive leukemia, there are actually 40% fewer GRD colonies than the control, which shows that it demonstrates that NF1 gap is reducing colony formation and growth. But we also saw that if we stimulated these colonies with growth factors, then there was no statistically significant difference between the number of GRD and control colonies, which shows that growth factors are rescuing this effect of NF1 gap expression. So in conclusion, because these leukemias are responding to NF1 gap restoration with reduced colony formation, we know that NF1 loss and RAS signaling are still implicated in AML maintenance. We also saw that growth factors are rescuing this effect of NF1 gap expression. And finally, in the future, we plan to repeat this experiment to confirm our results. So I just wanted to thank Michael Burgess, Eugene Huang, Jasmine Wong, Kevin Shannon, and members of the Shen Lab for a phenomenal summer. Also, Julia Ye, Lauren Meyer, Richard Shapiro, the Lowell Science Research Program, and UCSF for all of their support throughout. And I just wanted to say that I started this program 2 years ago not knowing any science, not knowing what a gene was, why it's important, what it does. And at first, reading my first scientific paper and learning how to present it was a struggle and very overwhelming. But with a lot of support from Lowell Science Research, I gained not only an interest in science, but also the confidence to believe in myself and tell myself that I can understand this. And 2 years later, I've researched at 2 different labs, last year studying the brain, this year leukemia, and I just really love how in science you can never stop asking questions. There's always new things to learn, and that's just been so exciting. And I'm so grateful for Lowell Science Research and UCSF for starting me on this journey of scientific discovery. So thank you.